I was telling my husband just a couple of days ago about this conference, and he works in healthcare. He's not a researcher at all. He's more on the working with community health centers around compliance for diabetes care and things like that. And he said this topic, risk stratification, is a really hot topic to these local clinics, an important one for them trying to deal with compliance. So it is a hot topic, not necessarily genetics, but risk prediction. So I want to thank Gina Peloso for helping me to put together this presentation. So if you go to the Framingham website, you see that we're interested in risk prediction. We even have a tab on the website called Risk Functions. And you'll see that we actually look at risk prediction for lots of different traits: coronary heart disease, several versions of that, diabetes, stroke, fatty liver disease, atrial fibrillation, cardiovascular disease, congestive heart failure, many of these. So Framingham's been involved in risk prediction for a long time. And it's had a long history in publishing on this topic. One of the first was by Bill Cannell, Dan McGee, and Tavia Gordon back in 1976. And they did separate calculations for men and women. They used sex, age, systolic blood pressure, total cholesterol, glucose intolerance, aka diabetes, left ventricular hypertrophy, and smoking in a logistic regression model. They were predicting CHD, the development of CHD, over 8 years of follow-up, and they found that the 10% of individuals at the highest risk category accounted for 20% of the events. So they felt that this was a worthwhile exercise. Kevin Anderson was one of the first in Framingham to bring survival analysis into risk prediction, and he used a parametric Weibull model and incorporating HDL cholesterol as well for predicting coronary heart disease. A well-known paper by Peter Wilson and colleagues used Cox proportional hazards modeling. They allowed you to consider either LDL or total cholesterol. We'll talk a little bit more about this a little bit later. And in particular, they used categories of risk. So what age range you're in, or what category of systolic blood pressure, and so on, and attached points to those categories so that people would add up the points and get their risk that way. And in fact, some of us older folks may remember seeing in magazines like Time Magazine or Newsweek a page where you could do this. They would have the point system. You figure out what your age was, what your systolic blood pressure was, and you do the point system right in the magazine. I don't think we have those anymore. So that was a point system, and that was frequently used for many years. Ralph D'Agostino and colleagues produced an important paper which said that the risk model that Framingham had been using worked in other populations as well, so long as you adjusted for a different baseline risk across populations. And so that was an important point. Ralph, again, and colleagues expanded risk prediction to looking at all CVD events, again using a point system. And then recently, I'm sure many of you are aware of the collaboration for development of risk prediction by the ACC and AHA. Came out in the 2013 guidelines initially, and more recently expanded in the 2018 papers. They were looking at hard CHD and stroke events, which they called atherosclerotic CVD. So here is the a figure from the Wilson paper. You probably can't see it particularly well, but up here, this is age, this is LDL or cholesterol. You had a choice of which of those you wanted to use, because not every clinic measured LDL back in those days. You had HDL. It had a combination of systolic and diastolic blood pressure, diabetes, and smoking. And you would figure out which of these categories you're in and get the points off of these little charts and list them here, and then sum them up and get your point total. And then you would go looking at these two charts to figure out your risk and how you compared to others. And those two charts look like this. So if you totaled up your points and you had 10 points, your risk was 11%. If you were using LDL cholesterol in your prediction model. If you were using instead cholesterol, your risk was 10%. And then you could compare that based on your age, and this points out, I think, that age is probably the most important risk factor for cardiovascular events. You could compare, if you were in the age range 50 to 54, you were at 10% or 11% risk over here, but the average risk for 10-year CHD was 8%, or the optimal, a lower risk was 7%. So you probably needed to do something to try to bring your risk down. So that was the Wilson paper, and this is sort of the way the point system worked. So coming back to the web page, the Framingham Heart Study, they also have, it's the same web page we were looking at a minute ago, but some of these are actually computerized. So one of them is looking at cardiovascular disease 10-year risk, and in this model, they were looking at these risk factors: age, diabetes, smoking, and so on. And you could actually do a risk score calculation right on the website. It's interactive. So the next, if you do this interactive, you can see that they have risk factors up here, what your gender is, your sex is, sorry. I think this is, I can't see it from here. What your age is, what your systolic blood pressure is, whether or not you're treated, whether or not you're a smoker, whether you not have diabetes, and what your HDL cholesterol is, and what your total cholesterol is, and then you can click on the Calculate button. And the Calculate button in this instance for this profile, this is a woman, 40 years of age, systolic blood pressure of 125, not treated, not a smoker, no diabetes, HDL of 45, and total cholesterol of 180. And that risk profile for the person for this woman, age 40, gave her a risk of 2.5% over 10 years. And the normal risk is also 2.5%, so she was right at the average. I guess normal is average. But the optimal risk was 1.3%. So we've done all these risk prediction models, but an important factor in doing risk prediction is evaluation of the performance of these models. If they aren't doing a good job of risk prediction, we might as well forget it. So I actually found this very nice whoops, pushing the wrong button. Let's see, go back. I found this very nice website at Columbia, which has a nice overview and discussion of risk prediction modeling and evaluation of risk prediction. So they mentioned that there are 3 steps. First, development, and in that context we use calibration measures. And the second step is internal validation, where we evaluate discrimination. And the third step is decision analysis. So in step one, the calibration, the question is how well do the observed events fit the expected model, or how well does the expected model fit the observed events? You certainly want that to be the case. If there's not a good fit, then your model's not very good. A commonly used statistic in this context is the Hosmer-Lemmerschau test, and asking exactly that. Do the observed number of cases fit the expected number of cases? And it's just a classic observed minus expected chi-square type of test. In the second step, in discrimination, you're asking the question is how well does the model discriminate or identify people who are going to have the disease or have the disease versus those who don't have the disease? Can you discriminate? And typical measures used in this context are the area under the ROC curve or the C statistic or the net reclassification index. And in the third step is decision analysis. How do and when will these predictions impact actual decisions in the clinic environment? So I should mention while we're here that these statistics have a lot of discussion in the literature, and I'm not planning to go into detail on these. We could spend half the day probably on this discussion. So I've just mentioned some of the typical ones used here, but you should be aware that there is a lot of controversy, discussion about these measures. So I went to the 2013 ACC-AHA paper by Lloyd Jones and others that presented these results of that effort, coming out of, I'm sure, quite a long, a year or more effort of trying to put together these guidelines for the ACC and the AHA. So they decided to look at atherosclerotic CVD, which was this hard CVD events like recognized MI, CHD death, hard stroke, and excluding transient ischemic attacks, for example, but both ABIs and hemorrhagic strokes, and stroke death. So any one of those events went under this label of atherosclerotic CVD. And the model that they developed, which is called this pooled cohort equation, came from data in 4 studies: the ERIC study, the cardiovascular health study, CARDIA, and the Framingham study. And they pulled the data and, using Cox proportional hazard analysis, came up with 10-year risk predictions, hoping that these would be used in the clinic. And on page S72 of that paper, way back at the end of it, they actually produce this display, and it gives estimates, or it gives regression coefficients, which are in this first column, or that column there. First for whites, and then for blacks, and at the top we have women, at the bottom we have men. And they are taking here now just a way to do it. So you have a person with different traits. So they looked at log of age. So you put the log of a person's age here. They have, can't see what that one is, but I know they are doing log of a lot of these risk factors, cholesterol and so forth. And they're looking at interactions and things like that as well. So it's a fairly complicated model, and not all terms fit in all of these 4 groups, but they did separate analyses of each of these 4 groups. And so what you do is you can, put the person's values for their risk factors in this column or a column like that, and multiply the regression coefficient times the risk factor to come up with the product, and then sum it up. And finally, because they were centering things, you subtract off the mean of those covariates, And you can use that to get the risk prediction. So for this woman with these traits and a baseline 10-year risk for white women of a number I can't see, 0.9 something, and the risk for this woman was, I think, 2.1%. And likewise, you have one for white men, and so on. So we used this model in the Framingham cohort to see how it worked in Framingham. Now, of course, we should say that Framingham was part of the development of this model, so perhaps a bit circular, and hopefully it will fit, but it's not exactly the same group of people that were used in the pooled cohort equations. So the ones we used were 4,921 subjects, ages 40 to 80, in the offspring at exam 5, or in the third generation at exam 1. We looked at this same atherosclerotic CVD event, and we did a follow-up of 10 years, as in the PCE equations. And we actually went through that exercise that I was just talking about of taking the regression coefficients, multiplying times the risk factors to get the sum, and then subtracting off the mean for each person. And we obtained risk estimates for each person. We also used those same, what we're calling PCE deviations, the sum minus the mean, in a Cox proportional hazards model. And this is what we came up with first for the estimates. So it's just a single estimate of risk for, we looked at females and males, and we combined them together. Well, and here we don't. You'll notice right away that those with events have higher risk, about 17%, versus people who haven't had events over the 10 years of follow-up, around 6%, and that men are at higher risk than women. I also note that there's a long tail of risk even among people who haven't had events. So, how does a genetic risk score function in the context of this model? So we went to a paper published by Nick Pay and a whole bunch of other people looking, this is coming out of the Cardiogram plus C4D consortium. They were using the 1000G reference panel, data were imputed to that. It was a case control study of around 184,000 subjects with 61,000 cases. They evaluated about 9 million variants, and we drew the genetic risk score from this paper, and we used selected the 57 variants that were significant either in additive or recessive models at 5 times 10 to the minus 8. And then we calculated the genetic risk score as a weighted sum of the risk variants, the risk alleles of these variants, using the regression coefficients from the additive model in the Nick Pay paper. So an interesting point that we observed here was that we have each of these bar charts is showing the spread of the PCE risk according to either high GR risk, intermediate, or low. So low genetic risk, intermediate, or high genetic risk. And you'll see that the traditional risk factors had virtually the same risk across all 3 genetic categories, suggesting that the genetic risk was independent of the traditional risk factor risk. We also took that PCE calculation and put it into a Cox proportional hazards model with 10 years of follow-up, and we found that it was highly significant, with a hazard ratio of about 2.4 for each unit increase in the sum in the PCE calculation. And then we also added the genetic risk score, as we mentioned how we calculated it in the previous slide. And again, we found the PCE deviation was significant, as well as the genetic risk score, both of them highly significant, with the genetic risk score adding about 2.2 times the risk per unit increase in the genetic risk score. So we also looked at some of these performance measures on this model. And so we looked at the calibration statistics, and we find, so one of the things you're looking for here is you don't want significance. Unlike most statistical tests, you do not want statistical significance to have good calibration. You want the data to fit the expected values well. And so we have good calibration. And if you look at the C statistic, if you have just the PCE calculation, it was about 0.78. Those of you familiar with the C statistics, 0.5 is like tossing a coin, 50% chance. So this is more than 50% chance, somewhere between 50% and 1. And when you add the GRS, it increases to 0.79, and typically, this has been my experience, that the C statistic does not move very much, even though that GRS risk score had a p-value of 0.0001. So, and that's one of the issues that people struggle with the C statistic. Going on to the NRI, we found that adding the GRS risk score to the PCE calculation had a statistically significant improvement in the risk prediction of this number, 0.097. So in summary, there's been a long history of risk prediction in Framingham and in other studies as well. Risk prediction, as I started out with, is commonly used in the clinic today. When I go to the clinic, sometimes my clinician will give me an estimate or different estimates depending on profile. And I'm told that the guidelines suggest that if you have a risk greater than 7.5%, you are to be recommended for treatment. And the ACC-AHA is hoping that these PCE equations are used in the clinic. We found in Framingham that PCE calculation, the 2013 estimates, fit well in the data and discriminated as well, with a risk of about 2.4 times per unit of PCE. And additionally, the GRS, the genetic risk score, also added significantly to the risk for atherosclerotic CVD, increasing the risk about 2.2 times. So I would like to acknowledge the Framingham Heart Study participants who've borne with us for many, many, many years. I'd like to thank the NHLBI for support, for more than 70 years of support, and the many researchers in Framingham Heart Study, especially Bill Cannell, Peter Wilson, Ralph D'Agostino, and Michael Pensina, who've been very much involved in risk prediction. Thank you.
Great, thank you, Adrienne. Questions? So please, Patrick. Were you raising your hand or coughing? Coughing. Early. Questions for Adrienne? Let me just ask, Adrienne, when testing these in non-European ancestry or non-white populations, what samples did you use to be able to compare?
You mean that Ralph did?
Yes. Oh, sorry, Ralph.
I don't remember all of them, but I know like some were, I think, a Spanish cohort. There might have been an African-American one, but I'm not positive about that. I would have to go back and actually double-check that, but they weren't all white.
Didn't Eric with an A, do some of this with Woody Chambliss and that? Yeah, but those were obviously small samples.
Yes, not the samples that we see in genetic studies these days.
Absolutely. Eric?
Yes, Eric.
Adrienne, thank you. Has Framingham's, obviously you did CHD, it's known as a cardiovascular cohort, but there are many conditions that are looked at. Has Framingham systematically gone down, pick your poison, the top 25 most common conditions in the U.S. and developed and implemented and tested and validated risk scores?
I think you've just assigned us a task. I think the answer is no. I think the ones that have come forward have been ones that particular researchers have been interested in, and so that's the way they've been developed rather than a systematic strategy on it. Yes?
So a quick question about the relationship between genetic risk scores and some of the phenotypes that are measured in the risk prediction models. So if the geneticists have it their way, many of these phenotypes have something to do with the scores. So this kind of adjustment makes it inappropriate because you can measure the genetic risk scores even before you manifest the phenotypes. So how do you deal with that statistically?
Statistically?
Or conceptually?
Well, I mean, you could get into Big Brother types of thinking. The babies are born, and you calculate their genetic risk score, and then you engineer their whole life. But I don't think we're going there. Methodologically, so I think what you're saying is that you concerns about biased estimates, perhaps, because you're looking at factors along the pathway. And there are some methods for dealing with that. We haven't directly here. Peter might have a comment, I see.
Yeah, no, I think the answer to Muin's question, at least for me, is thinking about the context. Like, how are you going to use this score? So in a setting where you don't have the exposures, then the genetics is what you have. But in lots of other settings, those will be hugely important and will sort of recalibrate, to use the word in more than one sense, your evaluation of the genetics. So I think actually what you've done is actually quite interesting, because I think aside from why do we need to collect this environmental data anyway, we just need the genetics. The other question is the other way around. We have the phenotypes, why do we need the genetics? And this is showing that each are, at least in this setting, each are contributing something.
Yeah, and I'd like to hear your comment, Peter, actually, on the fact that the PCE risk seemed to be independent of the genetic risk in these calculations, and yet you might think that some of those genetic variants are affecting some of those risk factors. So
my brief comment is sticking with the empirical fact that when you put that they seem to be independent. My gut feeling is that the mediated effects of genotype through these phenotypes is subtle and not a game changer.
Okay.
Like, it's important to adjust for these things. Like, you can see an attenuation of the family history effect, kind of obviously. And BMI for diabetes, probably there's some attenuation, but again, the mediated effect, I suspect, is going to be moderate.
Yeah, no, good point.
Yes, a comment following up on this independence. So you show that the absolute risk, according to the PCA, was not different depending on the genetic risk
Yes,
scores,
that's right.
which you're calling independent, but it's basically a multiplicative interaction. It's sub-multiplicative interaction, right? Because if they were multiplying, you will expect to see the absolute risk to be higher in the high, which is what we
Well,
see in
yeah, the
cancer.
absolute risk of both of them would be higher.
Yeah.
But the PCE risks seem to be the same regardless of the genetic risk score.
In the
Yeah.
in the absolute
Yes,
scale.
yes.
So in the
On the
relative
absolute
risk,
scale.
in the
Yes.
relative risk,
Yeah.
so that's actually different than what we see in cancer, where we see that the absolute risk is actually higher in the high genetic risk. So that's interesting.
Mm-hmm. Yeah, it is.
Cardiovascular.
Great. And
I could just.
oh, please, Naomi, and then Bob.
I was just going to add to this discussion. I don't know if you've seen the Mike Inoue paper that was published last year in the Journal of American College of Cardiology, and that was using the UK Biobank data. Okay. I was just suggesting looking at the Mike Inoue paper. I don't know if you know that one.
No.
It was published last year, so it's used the UK Biobank data with coronary artery disease and looks at the environmental and genetic risk factors together. It's a very nice paper.
Did it find a similar thing, or did they even look at that?
Well, then the environmental, because of the correlations, each risk factor independently explains a certain amount, and then together they do explain more, but they're correlated. So it's just a bigger data set for evaluating.
Yep. Thank you.
To Muin's question, I do think it's important to think about sort of we think about this as identifying those at high risk and being able to treat those effectively, but it may be that the genetic risk score may add something to identify people who have a high Framingham risk, who have a low genetic risk, who we can actually relieve of the duty of all the interventions. And that's important to think about in terms of
Yes.
utilization of healthcare.
Absolutely.
Any other questions for Adrienne? If not, we will have a discussion time.
Right. So this is like an editorial comment, but I'm sitting next to Terry, so I'm allowed to make those comments. And that is, as we go through the day, there'll be a focus on coronary disease because of the people around the table, and because that's an easy phenotype it's an easy phenotype because there are lots of it. And but there are other phenotypes that in which this kind of approach may yield different results, and I look forward to hearing what happens when you apply this kind of thinking to lots of other diseases, lots of other acquired traits, and I hope we'll hear about that today.
Good.
I'm sure we will. All right.